Good afternoon, boys. Thank you so much for joining the session. Inshallah, we will try our best to make it fruitful and you are ready for your exam. And inshallah, Allah Kareem, you score good. Now, I will share my screen. Every one of you, please log into ClassKick. We will be using ClassKick for today. Make sure you log into your account. Okay, when you log into your account you will be able to uh, see.
Hi, Mr.
Hi, good morning. I will take attendance after 5 minutes, like at 1:10 I will take attendance. So now you log into ClassKick, please. So today is our second session of the SAPET 09 10 20 21. Okay. On grade 11 together. Okay, so let me just take quick screenshots and record. Everyone please log into the ClassKick. Thank you so much. Okay, once you log into the ClassKick you will see there is a sheet that is called as practice sheet for CA1. Anyone please confirm me, do you see this sheet?
Yes.
Yes,
Okay,
Mister.
very good, very good. Open it please. We will try our best to finish it because um, this is very important to have a practice, and we will try to finish and link all the concepts possible. Let me open ClassKick on my iPad, laptop, tablet. Okay, here. Okay, in the meanwhile you open it and you go through it. I will just go to my
Mister.
name. Yes.
1.5. It's not including in
No, no,
CA.
no, no. 1.5 not included. ENI.
Okay.
Okay, so I will reflect my own screen. ENI from ENI. Practice sheet. Everyone open practice sheet. You see this one. It says that grade 11 bio 61 practice. Okay, so definitely we need to figure out these. Please, everyone, because separate session always we consider it as your marks also. So you need to see, for example, if there is any chance of the bonus marks, they are considered on the separate, and that is only possible if you finish all the points. Yani if you finish all until wherever I will finish. So if you join, it means that you are supposed to answer all the questions. You keep trying answer the questions. In the meanwhile, I will put attendance. Okay, so I'm going to take attendance, and you keep, so please keep saying me your sections. Hamad Abdullah.
Yes, Mr.
Hamad Abdullah is from which section? uh, ENI.
Nice. Yeah.
Okay, yes, got it. Then it is Hamad Sultan.
Mr. CAI.
CAI. Okay, very good. Next, please keep saying Hamad Sultan. Yes, okay. Hamdan Ahmad. ENI CAI.
Yes, ENI, CAI.
Yeah, now some of you I remember actually. I will try to make it fast. Hamdan Muhammad, CAI?
Yes.
Okay. Next, Hazza Saleh. No, Hazza Umar. Sorry. Hazza Umar, CAI.
Yes, Mr.
Hazza Saleh, ENI.
Yes, Mr.
Okay. Hazza Saleh, ENI. Okay. Next, Hilal, ENI.
Yes.
Hussein, CAI.
Yes, sir.
Hussein, CAI. Okay. Next, Khalid Nasser.
Yes, Mr.
Which one? ENI. Okay. Mansoor Talal.
CAI.
CAI. Okay. Next, Muhammad Ghanim.
Yes, Mr. Here.
No, no. When I call your name, say me your section.
What, Mr.?
Section. I section. ISAF.
ENI.
ENI. Okay. Muhammad Ghanim. Okay. Next, Muhammad Ubaid Hassan.
ENI.
ENI. CAI.
ENICI.
Okay. Muhammad Saeed Ghulam.
ENI.
ENI. Sorry, boys, this is really very important to make a full record of you. We are about to finish. Mubarak Fahad.
Yes,
ENICAI.
yes, Mr.
Okay. Next, Rashid Khalifa. ENI.
Sir, CAI.
Oh, CINI. Okay, Rashid Khalifa, yes, here. Salim Abdullah Rashid.
ENI, Mr.
ENI. Okay, Salim Abdullah Rashid. Okay, Salim Muhammad.
CAI.
CAI. Okay, and Turkey. CAI.
CAI.
Okay, anyone I missed you? No, all 18 I did because there is no one else joined. Okay, we will now move back to our topic. Boys, there are 4 topics which are coming in your exam, and these are the first 4 topics. If you look at my screen, only 1.1, 1.2, 1.3, 1.4. So remember that these 4 topics are only coming in your exam. Don't prepare 1.5 for your CA. Okay, topics for CA1. Definitely next topics are coming in your end of term, but for the time being you have to prepare for the CA. After that, the first topic is Mendelian genetics, where we studied about genes, allele, gamete, phenotype, genotype, heterozygous, homozygous, hybrid, and monohybrid, dihybrid, law of segregation, law of independent assortment, all these things. So we have to see the questions related to them. So today we will do the questions and we will keep practicing. So you better keep answering side by side. Yani when we go to the question, so keep answering. It will make things easier for us to understand, and also you will be knowing that what is known to you already and what is not known. Okay, so let me just do this. Yes, so that I will be knowing who is answering and correctly or incorrectly. But it doesn't matter even if you have a confusion, just keep asking. Question number 1, boys, ignore the numberings on the side because the questions are taken from the previous exams for the practice. So just these are the, that's why they have different numbering. We don't care for the numbering. The first question says that during gamete formation, remember that when we talk about gamete formation, what is actually, this is capital T, this is capital T, small t. This is a gene. Okay, this is a gene. So when we say that gametes form, actually these alleles separate. These alleles will separate, and you will have one capital T and one small t. So this is one gamete, this is another gamete. Okay, so this is what during gametes form. What process allows the separation? Now when we talk about the separation, you should be able to know that, like, which of the processes below talks about separation. Yes.
Sir, segregation.
Segregation, Mr.
Segregation, very good. We studied that segregation means the separation of allele. Okay, take the mouse and press D. Okay, we'll take the mouse and press D so that we grade it. So D is correct answer, and on the top you see our one question out of the two is correct. So that's why 3 marks out of the 6 marks are given. It's auto graded. Okay, now we move to the next question. Give me a minute, sorry. Okay, now in the next question down we have, they say that what is the phenotype? What is a phenotype, by the way? Define.
The physical appearance.
Very good. P for phenotype, P for physical appearance. So it means that you are going to just say color, just color, okay, not the letters. In the phenotype we don't use letter. It means that option A and option B can never be correct in this answer because they are using letters. Okay, so these two are the genotypes actually. Okay, these are not the phenotypes. Now, so what is the answer for this question?
D.
D, very good. That is the white. Okay, so we take the mouse and answer D, and it gives me that, because I'm using actually my teacher's screen, by the way. The screen which you are using is a teacher screen. That's why it shows me green. It doesn't show you green. Okay? Fine, because I'm using two screens. We will move to the next question, please. By the way,
Yes, Mr.
yes?
The last question down
Yeah,
below.
yeah, I'm going to read it. Sorry, I missed it. In the image above, that's this one. So in this image, the white trait is dominant or recessive. What is
Recessive.
white trait? Recessive. Very good. Circle it. And the purple trait is
Dominant.
dominant. Very good. Okay, let's move now to the next page. Fine. Here they say you label the image. Actually it is labeled, you by chance, when the picture was kept here, it is labeled, but I will explain you what does this mean. So first of all, you need to know that what is this whole picture, yani what is the name of this. I'm talking about this option actually. What is the name of this whole picture?
Chromosome.
Very good. These are chromosomes. Okay. Now, in the chromosome, what is most important to see? These things. Like there is a white flower, then this part code for white flower, and this part code for the purple flower. Okay. The location the location where the genes are present, which are labeled over here, this and this one, these are the locations on the chromosome where these alleles genes are present. So what we say them, what is this location called?
Locus.
Locus. Very good. But since we are labeling two, because one arrow is coming from below, one arrow is coming from up, so that's why we say it as it is a loci. Loci is plural. And the because this is a gene, this area is a gene, but they are in pair. One side code for the purple color and the other side code for the white flower. It means that it looks like capital P small p. Okay, it looks like capital P small p. This is a gene, but what is the name of this p and what is the name of this p alone?
Alleles, Mr. Alleles.
Alleles. Very good. So that's why you can see on the diagram they have labeled it as allele. Okay, is that clear? Yes.
Mr. How you know that one is dominant and one is recessive?
Okay. Sometime it's very common to you. Yani we can use our own knowledge based on the colors. You can say that white is always recessive. Okay? Yani this is very general. White is always recessive. So whoever comes with white will be dominant. This is number one. Number two, you will be given some options. Like if we, let's say, go back to the previous question, and you see over here in this diagram, they have shown you that this is purple, this is purple, this is purple, and this is white. So from here, you can identify who is dominant and who is recessive. Okay?
Okay, Mr.
Now, next question, please. Which genetic cross gives you 1:1 phenotypic and genotypic ratio? Okay, now without think any without solving it, actually you
A,
can.
Mr.
It should be what?
A.
Did you try to solve it? Sorry, I couldn't see your name. Did you try to make a Punnett square for this? No, you did not make Punnett square, and you just answered. Boys.
Mr. 121.
Yes, you need to do the Punnett squares, boys. Okay, it won't take much time from you. Quickly do the Punnett squares like this, very small. Capital T, I'm taking A here. Capital T, small t, capital T, small t. And it will come what? These answers will come. So you have 1 TT, 2 TT and TT, and 1 small t, small t. So this ratio is coming 1:2:1. And this is the genotypic ratio, by the way. But these will give you 3:1 as the phenotypic. So their ratio is coming different. So A is not an answer. Let's go for B. So you have capital T, small
B
t,
is
small
correct,
t.
Mr. Right.
B. But you got all capital T, small t. How B is correct? You need the ratio 1:1. This is B. B is also incorrect. Now we will move to C. Okay, and the C says capital T, small t, small t,
Mr. The
small
answer
t.
is C. I try it.
Very good, Salim. Yes, the answer is C. So you get two capital T, small t, and two small t, small t. Two will cut with each other to 1 times 2 to 1 times 2, and it will become 1:1 automatically. And there is no option 1:1 capital T, small t, and 1:1 small t, small t. So the genotype and phenotype ratios are same. So this is C. By the way, I have boys. Yani, if you really want to score high marks, don't rush. Don't rush. Your exams are not timed. Okay? That the latest information which we received, and you will receive also, your exam will be open for 40 minutes. It is a 40-minute exam. It will be open for 40 minutes, and you have enough time to answer. So please don't rush. Stay on the question. Try your best to answer it 100% correct. Use the practice as much as you want. Yani you need to do all these Punnett squares, do all these Punnett squares. Get the answer and then choose in the exam. Maybe you don't have an option to go back. Yani I'm not sure what the latest about the go back.
Oh, Mr. They sent the last mail. We have the option to move from question to other question.
You can move back. No, forward okay, but back.
No, Mr. They told us that we can.
You can.
Yes.
Okay, if you can, make sure if you can, then only, yani you if you are thinking you are spending a lot of time on a question and it doesn't work, choose the answer which is correct according to you, but make a note with you. Yani question number 4 I have to review. So when you finish all other questions, go back to question 4 and make sure you do it correct. Boys, each question really makes a big difference on your marks. So yes, Mubarak.
Mr. An exam, can you bring paper? Yes.
I think so the teacher can give you, but keep one blank paper next to you and show the teacher, Mr. or Miss, this is purely blank. They will say if it is allowed or not allowed. Okay?
Okay, Mr.
Yes,
Sir,
yes, I'm done.
what you did after you found that there is two of T, capital T and small t, and how you crossed, you got one.
Okay, because this is 2 and 2, simplify them as per your math rule. Yani for example, Hamdan, if it is 10 ratio 10, if 10 ratio 10, we can simplify it by saying that 10 1 time 10, 10 1 time 10, so it means that they are 1 ratio 1. You got my point?
Yes, sir.
Yeah, same goes for if it is 2 ratio 2,
Mister,
simplify them. Yes.
if it's just give us one phenotype, it's mean 100%.
Yes, yes, that's what happened in option B.
Yes.
Yeah. Okay, by the way, boys, remember, 1 ratio 1. No need to work all these if you know it already that this is a test cross. Yani, what we did right now is a test cross. 1 ratio 1 always comes by test cross. Okay, and the test cross is always made between a heterozygous and a homozygous recessive, always. Yani if you learn this hint, no need for you to figure out below. Yani no need for you to try all these to get the answer. I'm saying again, listen. 1 ratio 1 is the ratio of a test cross, and a test cross is always made between a heterozygous and a homozygous recessive. Okay, if in the exam you get a ratio, I'm not sure, just give me 1 minute please. If in the exam you get a ratio and they say you which of the following will give you 1 ratio 1 ratio 1 ratio 1, now it's 4 ratios, but all ratios are 1. Again, this is a test cross. This is a test cross, boys. And test cross, I told you, this is made between a heterozygous, okay, it is between a heterozygous dominant crossed with homozygous recessive. Okay, now because they are 4 ratios, we can say that this is a dihybrid cross. You remember the ratio of the dihybrid cross was 9 ratio 3 ratio 3 ratio 1.
Yes.
Now this is the dihybrid ratio, and this is the test cross ratio for the same dihybrid. So what we are going to do now, we will do the same thing. We will put a heterozygous and we will put a homozygous recessive. So heterozygous will be capital R, small r, capital Y, small y. So this is a heterozygous, and multiplied with yani crossed with homozygous recessive, so small r, small r, small y, small y. No need for you to figure out all the options given. Just search for one heterozygous and one homozygous recessive, same as monohybrid cross. Search for capital R small r crossed with small r, heterozygous with homozygous recessive. Is that clear?
Yes, Mr.
Okay.
Sir,
Again saying, again, uh, 1:1 is always known as a test cross, and a test cross is always between a heterozygous generation crossed with a homozygous recessive generation. Okay? No need for you to figure out all these. Okay, I'm going to write here the 4 types so you can easily choose the answer. Okay, yes, Hamdan?
Mr. How I know if it's a dihybrid?
Because okay, Muhammad Ubaid, can you tell him how to know if it is a dihybrid or monohybrid?
Yes, sure, Mr.
Hmm.
Mr. Monohybrid, if it's one, two, or if it's one, dihybrid, if it's two or more.
Okay, let me simplify what Muhammad Ubaid said, Hamdan. You can choose. You can easily know if it is a monohybrid cross or a dihybrid cross based on the ratios. You need to look at these ratios. Okay, these ratios. You need to look at these. These. If the ratios given to you are 4, it's always dihybrid. And if the ratios given to you are either like 1:1, 3:1, 1:2:1, all these ratios are, sorry, I'm writing above. Okay, all these ratios are monohybrid, and all these ratios are dihybrid. You, yani, when you make your
Mister,
notes,
can you go down?
where here?
No, finish. Thank you.
Okay, so you need to memorize these ratios, boys. Write them somewhere. 1:1 is a test cross ratio for the monohybrid cross. 3:1 is a phenotypic ratio of the monohybrid cross, and 1:2:1 is a genotypic ratio of the monohybrid cross. And these ratios, 9:3:1 is a phenotype and genotype for dihybrid, and 1:1:1:1 is a test cross for the dihybrid.
Mr.
Yes?
What's the ratio of it was homozygous dominant with recessive homozygous?
Homozygous. See, you focus at your words. Focus at your words. Homozygous dominant with homozygous recessive. Sir?
Yes.
It will give you 100% like this one. Look, in question above which we solved, you can have a look over here. The answers of option B. The answers of option B. This one. This is a homozygous dominant with a homozygous recessive. And what we said, all are heterozygous. So 100% heterozygous. It will be same for if you are doing it as a dihybrid or monohybrid. Doesn't make a difference. You will always get 100% heterozygous. Okay, let's move further because we took a lot time here. Question 2, please. A dash is a segment of the DNA that controls the production of protein.
Right.
Locus is the point where gene is present. Trait
B,
is
genes.
a character that you can see. Alleles are the alternative forms of a gene. So what is a segment of the DNA? Gene. So answer B. Okay, let's move next. Okay, now here we have fill in the blanks, and we are going to, boys, if it is hard for you to type into the blanks, make your zoom in your screen, okay, so that you can make it big and you don't touch here and there different places to answer. Okay, the first terminology, alleles of an organism has for a trait designated by letters. Focus on the word. What he is saying?
Genotype.
Genotype. Bravo. Okay, quickly type, boys. We are not going to stay here for long. Okay, take mouse and enter your answer into it. Wait, I need to check what's happening with me. Okay, quickly, boys, please. Okay. I know. I think so. We need to take mouse, click in, and type genotype. Okay, fine. So my answer is genotype, and I got 3 out of 3. Okay. Heredity units existing as segments of the DNA on the chromosome. Just now we did a multiple choice.
Gene.
Gene. Very good. Okay, so we are going to the next option, and we are going to take mouse and write here as gene. Okay, next please. An individual having two different alleles, two different focus on the words.
Heterozygous.
Very good. When you focus, yes, when you focus on the words, you easily get the answers. Okay, heterozygous. Okay, different forms of a gene. Allele. Very good. Excellent, boys. Amazing. The visible expression or the physical traits. Phenotype. Excellent. Phenotype. Okay, the last one. An individual possessing two identical.
Homozygous.
Very good. Sorry, I was drinking water. Yes, this is homozygous. Okay, let me type it also. Homozygous. Okay, we are done with this. Huh? Why it's giving me wrong? What happened with homozygous? Wait 1 minute. What happened? Let me try capital HOMOZY homozygous. Huh? Still giving wrong? 1 minute. It should not give me wrong, because I don't want you to lose the marks. Let me just quickly check the answer key. Go back. Edit. We are on which slide? Slide number 3?
3, Mr. Yes.
Yeah, let me check this. By mistake I wrote all wrong. Wait. Homozygous. Because some of you write capital, some of you write small, and this was for 3 points. Okay, please rewrite the answers for you. Okay, or maybe if it is written already, it should give you correct. Wait, let me go down and check for me, please. All, I'm saying again, all of you answer, because if I don't see your answers, yani this is no use to join the session. Okay, homozygous, it's correct. According to Mendel's principle of dominance, if a pea plant is homozygous dominant, so answering such questions, you can definitely circle and write homozygous dominant. It means it is capital T, capital T. Sorry, why small t?
Tall dominant.
Yeah, phenotype will be what? Tall.
C, tall dominant.
Tall.
Okay, and because the dash allele will show up, the dominant allele will show up. Very good. So
Sir.
answer is C. Yes.
But Mr. It's being like dominant, homozygous doesn't necessarily mean that it could be tall. Like short phenotypes could also be dominant.
No. Yeah. Yes, you are right. Short can also be dominant. Yes, as in human height is dominant, and dominant is the short height in human. Yani if someone is very tall, like 8 feet, 7 feet, 8 feet, 9 feet, so they are recessive. We rarely see them. Actually, recessive mean we rarely see them. So in human, the height of the human the short height in human is dominant,
Yes, Mr.
and the tall height in human is recessive. Yes, you are right. But don't go into those details. But you have the concept. I'm happy for that. Okay, we will quickly go here. Capital B, small b is homo or hetero? No need to write because no marks for here.
Hetero and
Hetero. Then
Dominant.
what will no what we'll say? Yeah, dominant. Okay. Capital B is what?
Homozygous dominant.
Dominant. Very good. And small b
Homozygous recessive.
very good. Okay, let's go to next question. Here in the rabbit, R is dominant to small r. The cross shows that a cross between the rabbit with the dark fur and the with the white fur. So you have one black, one white. You cross them and you get two black, two white. Yalla. Now, two black, two white. Remember what I told you in the previous discussions. It is 2:2. Cut them. What you get? 1:1. So it is a test cross and it is a monohybrid test cross. Sorry? Very good, very good. So it is between a heterozygous and a homozygous recessive. Yes, the answer is A. No need to figure them out all. If you have the concepts, you can apply your concepts. You see here you have two black, okay, you have here two black and you have here two white. Put them in a ratio, cut them, you get 1:1. It is a test cross and it is a monohybrid test cross that is always between this A option. Okay, no need to figure all four together. And of course, one more thing, boys. Here, see, the three other options are very easy to get, they are wrong. Very easy. The fourth option is not possible at all, and the other two options will give you all dominant because they have capital R on one side. So you never get the recessive. Okay? Next question. A short-toed animal was crossed with a long toe, and all the offsprings had short toe. All of them had short toe. One of these offsprings was crossed with another long toe of the same species. Which ratio? So how you figure it out, I will give you a hint so that next time you do it by yourself. A short toe, which is, let's say, small t, is crossed with a long toe, which is capital T, and all the offsprings has the short toe. By the way, here he is taking short toe as a dominant. He is taking actually short toe as a dominant, so we have to replace them. Sorry, he didn't tell you, but you can get it from here from the result. When he crossed, he got all the offsprings as short toe, so it means that short toe is dominant. So short toe is small capital T, small t. All of these you got short toe, so capital T, small t. One of these is crossed with another long toe. So this is long toe. So you cross capital T with small t. Yalla. What will be the answer?
1-1.
Very
1-1.
good. Very good. Again, you got one heterozygous with one homozygous, and that's same as previous question. And that is the ratio is 1:1. So answer is A. Very good. Let's
Mr.
go to the,
sorry, can you explain it again?
okay. How you know that he did not tell you which of these is dominant and which of these is recessive? He did not tell you. Now this is your smartness to choose who is dominant and who. Actually, by the words short and long, you may think that long is dominant and short is recessive. That's what I did. But when I reached here, all the offsprings had short toe. From here, you got it, short toe is the dominant character. So what he did actually, I will show you what he did. You need to write like this: short crossed with long, and all offsprings come short. So from here you can get who is dominant. You got it?
But Mister, how you when you test cross them T
Okay,
T capital
okay,
and T
okay,
small?
okay. See, now you put their genotypes. You can put their genotypes, any genotype, SS or LL or TT, doesn't matter. So you put their genotypes. So of course, this should be the one you have here, short one, it is always capital, capital. The long one is small, small. And this one is heterozygous. Now, he says that one of these offsprings, yani one of these, crossed with another long toe. You know this is the long toe. So you cross capital S, small s with small s, small s. Clear?
Yes, Mr. Clear.
Okay, let's move next. Salim, is that clear, please?
Yes.
Okay, question number 6. Ignore 4, it's incomplete. Question 6. In one of the type of the plant, the allele for the red flower is dominant. By the way, in the previous question, in question number 2, he did not tell you who is dominant and who is recessive. You worked it yourself. But in this question, he is telling you. So it's easy. A plant with the red flower is crossed with a white flower. Same thing. Choose, let's say, same. SS, SS, and SS. Replace them with RR. So you got all red. Half of the offsprings have red flowers and half of them have. By the way, a plant with red flower is crossed with a plant with white flower. So 50-50. Again, boys, this is same test cross. So what's the answer?
D.
D. Very good. Any doubts about this question, please?
No.
Okay. Next question. You have
Mr.
this cross. Yes.
Can you go back to the question?
Yeah.
Mr. know if it's homozygous or heterozygous from the offsprings, yes?
Yeah. You see half of the offsprings. When it says half of the offsprings, it means that one of the parent above is heterozygous. If he says all of the offsprings, if he says all of the offsprings are red, it means that one of the parent is homozygous dominant. Okay, let's move next to this cross, boys. Here he says that the diagram shows you some cross between the two wolves, and one of the wolf is brown, while
Mister,
yes.
before we start, C and D are not possible.
Very good, excellent. So C and D are not possible because he is flipping them. He is writing genotype as brown, which is not possible. Sir? And also A is not possible, boys. Why A is not possible?
Single allele.
Yeah, because this is a single allele. It's a gamete, not a genotype. So automatically, without struggling all this, yani without struggling, what is happening here? Who is this? Who is this? Blah blah blah. Just look at the options and cross them out. This is wrong. This is wrong. This is wrong. Only this is the correct answer, and that's it. Move to the next, please. Okay. Okay, boys, the Punnett square has been done for you. According to the Punnett square given, what is the probability of the dominant trait among the individuals?
1/2.
Very good, excellent. These two are the dominant. So this is 2 out of 4. Again, simplify it. Do 1 times 2, 2 times 4. Your answer comes 1/2. Okay? So this is 1/2 and the answer is B.
Sir, it's a 50-50 chance.
Yes, bravo. 50-50, yes. Okay, next question. What is the phenotypic ratio observed in F2 generation? Phenotype F2.
3-1.
Very
3.
good. 3:1. This is what you have to remember. Yani nothing to do over here now. Okay, let's move to the next page. If a brown, you need to do this Punnett square and find the ratios, and we had done this a lot. This is again a test cross, heterozygous, and you are taking a homozygous recessive, this is a test cross. We have done so many test cross on the back. I'm not going to do this. You can try it yourself, okay? Let's move to the next question. Which of the following statements are true about the Gregor Mendel and his importance to the world of the biology? He is the father of genetics? Yes or no?
Yes, Mr.
Yes.
Yes. He explained you the inheritance?
Yes.
He did.
Okay. And
Yes.
he explained how dominant allele mask the recessive allele?
Yes, he did.
Yes.
Oh, all of the above. So it is D. 1, 2, and 3. Okay. Mendel used only dash. Which one? Hybrid or true breeding?
True breeding.
True breeding. Very good. Plants in the parental generations because it was produced the same. So he used the true breeding. Let's move next. So let me just circle this true breeding. This one. Okay. Gregor Mendel contributed to genetics by concluding that traits are not inherited from one generation to other, inherited to the next generation, only expressed in some specific generations, and characteristics that come only from the pure breeds.
By next generation.
Yes, inherited by next generation. Very good. Gregor Mendel is known as the father of genetics because he is the first one to conclude that
From parent to offspring.
inherited from parents to offsprings. Very good. Okay, next. Gregor Mendel used pea plant in his study because pea are true breeding, meaning that they
C, Mister. Offspring with multiply forms of a single trait.
did we
No,
study
Mister. B. B.
B? Yes, not C. We didn't. Mendel didn't say multiple things. He just said one trait. Yani offsprings with only one trait. So this was his concept. Multiple was complex pattern that we are studying now. Yani which don't follow the Mendel. Okay. What process is used by Mendel to cross male and female gamete?
Cross
Cross-pollination.
pollination.
Very good. It was the cross pollination. Okay? Not cross fertilization. He did cross pollination.
Sir,
Yes?
can you explain question 3 again?
3.
Can you go up? Yes.
Mendel used only dash parents in the parental generation. Okay. Parental generation, Hamdan. What do we mean by parental generation? The parents, the P
Yes.
generation. Okay. Whenever Mendel starts in P, he says that, like, let's say, tall crossed with short. This is what he always said. And then he said, tall is capital T, and short is small t. Always. Remember in the P. And all these are the pure breeds, not the hybrids. He gets hybrids when he cross them and he gets capital T, small t, and this becomes F1 generation. And F1 generation is a hybrid. Okay, and this is called as hybrid. So hybrid was a product of P generation. Okay.
Mr. But how they say that it produced the same trait in the offspring, so it will
Yes.
be
Yes,
thinkable.
no. See. Trait is always that you can see. So what is capital T, small t will show you. It will show you tall or short?
Tall.
Tall. Okay. This is number one. Number two, it's not getting anything different. It's getting one T from here and one T from here. So it is showing the same trait. One capital T, one small t. It is not showing you different traits. Okay? 1 minute, please. Okay, boys, let's move next. We have still 3 pages. Now we are jumping to topic 2, 1.2, genetic recombination and gene linkage. What do you see in this diagram, boys?
Crossing over.
Crossing over.
Okay, some of the questions are very straightforward. You don't need to struggle with them. You don't need to fight with them. Simply keep answering. Okay, next. Use the crossover frequency, which is AB is 40, sorry, AC is 45. So you will go for, first of all, choosing the options that stand with AC in front and last. Which option stand for A and C?
The third one.
There is only one option. No need to figure any other thing. You got my point, boys?
Yes, Mister.
Again, I'm repeating, because such questions take a lot of your time. You need to work with them. Yani you can work all. Draw a line, put A here and C here, and put it as this whole is 45. Cut it from here. Then AB is 40. Put B here and say that this is 40. Cut this one. Then BD is 30. So you have to move back in this direction, because this is total 40. So somewhere here is BD, and this distance is 30, and you have BC, which is 5, because A to B is 40, so B to C is automatically 5. So 40 plus 5 is 45. You need to figure this all, but I gave you a hint which can work and cannot work. Yani this is different scenario. But in the
How, Mr.
question now, it is working. What is your first allele? A. And what is your last allele? C. Okay, halas. Sorry, gene, not allele. So look over here. You have A and D. This is not answer. You have B and C. This is not answer. You have A and D. This is not answer. So only answer which is available with A in the beginning and C in the end is C. You got it? Without figuring out all this, we have answered it before. But I did this all for you to understand. If, let's say, this answer also says you, yani A to C, let's say, the first one. So now you have two answers, this and this, to choose one. Then you can go next step, next step. Wherever you figure your steps, leave it. Don't go into all the steps. Okay? So you can save time in your exam by this. So answer is C. Let's go next, please. You have 4 pairs of the chromosomes. Okay, and if 2 houseflies are crossed, look here, this is very, very important, boys. When he
16.
says that 2 houseflies are crossed and you have 4 pairs. By the way, pair mean how many total chromosomes?
8.
8. So 4 pairs mean 4 times 2, because pair is always 2, is equal to 8. This 8 is equal to 2n. 8 is what? 2n. You need n.
So Mister, it's going to be 8 to the power of 2, which is equal to 64. So our answer is B.
Yes, you are correct. But first let me figure out this n. N is what? 4.
4.
Okay. Now you need to do it for one first. Okay, so first one fly. So 2 to the power of 4 is equal to 2 times 2 times 2 times 2. So this is answer for 1. 4, 8, 16. Okay. Now what he says you in the question, he says that 2 are crossed. So you need to take what? Again, same answer.
Another
and then
pair.
Yeah, another pair. So 16 times 16. What answer will come?
64.
La,
256.
64.
256.
16 times 6, 256. Yes, 256. Okay. So this answer is C. So your
Sir,
answer
can you
for...
explain it again?
Okay, give me a minute. C. Let me first put C as an answer. Okay, it is correct. Boys, listen. This is very important question, because you will definitely have a question related to it in the exam. Okay, I'm going to undo all. Wait, let me just undo to get space here. I don't have eraser to erase it. Wait 1 minute. Ah, I have. Okay. Let me quickly erase it. Okay. And my answer was C, so that I don't forget. Okay. Look, boys. Here I erased everything. In the question can be of two types. Sometime he says you 4 pairs, but he will not say you 2 houseflies are crossed. No, he will just say you how many crossing over possibilities are available in this one. So you need to do it only one time. But in this question, he says that 2 houseflies are crossed. When he says 2 houseflies are crossed, so you need to do this twice. Okay, so first of all, you have 4 pairs. That means 4 times 2. That means 8 total chromosomes. 8 are the total chromosomes. You need to half of the total. So that is 4. So your n value is 4. And you have a formula to find the combinations. The formula is 2 to the power n, and that is 2 to the power 4, and that means 2 times 2 times 2 times 2, and this answer comes as 16. If it was only one butterfly to find the answer in this question, it would have been A. The answer would have been A. But he says that 2 houseflies are crossed. So this answer 16 is for one housefly. The same answer will come for the second housefly. Now what you will do? 1 multiplied by second, so 16 multiplied by 16. So this answer is for one housefly, and this answer is for the second housefly. So what answer comes now? 2, 5, 6. By the way, you will be having calculator in your exam, but that will be maybe a simple calculator. I don't know. I have never used the calculator in the Swift Assess. So you can have an extra calculator with you, scientific calculator, so that if you have to take a quick power like this, you can easily take it 2 to the power 4. Otherwise, you have to do it manually. Is that clear? Can we move next?
Yes,
Mr.
Mr.
Mr. 256 is the number of fertilized eggs types
Yes,
of 6.
yes, yes. Is the possible type of the fertilized eggs, yes. Boys, when they say you 2 are crossed and they say you fertilize the egg, then only you need to go to this step 16 times 16 or whatever answer come. Okay, but if they don't say you for the fertilized egg or the 2, they only say for this housefly only, then you have to stop until this 16. That's why in the exam you have both answers. You have 16 as an answer and you have 256 as an answer, because always such questions have both answers there. You got my point? To trap the students, because some of them they will reach 16 and they will say, oh, answer is 16. They will not go further. Next question, please. Here,
Mr. Can I answer this?
64.
yes, please.
Okay, so Mr. Just like we talked about in the last question, we don't have any houseflies crossed, so we're just gonna do what we did, 2 to the power of 6, and our answer is gonna be 64.
Very good. So we are not extending here until the second. We will stop at the first level. Okay? But because it is here, it is saying gametes, only gametes, not fertilized gametes. It's saying only gametes. Okay? He's not saying you that 2 houseflies are crossed. And it is 6 pair. So it means that remove the pair, by the way, you only get 6. By the way, the question with the pairs is the easiest question. You can take the first number, because if it is 6 pair, 6 times 2 is equal to 12. Okay, and then you again cut with 2, so you get the answer again 6. So always directly take 6 and ignore pairs. But if they give you a question like this, now see, a housefly, I'm writing here. Okay, a housefly has 48 chromosomes. Okay, how many how many possible this, whatever. Now here what you need to do?
2 power 23.
Divide 2.
Yes, yes. What divide 2?
48 divide 2.
Yes, here you have to take 48. You need to divide it with 2 and the answer comes 24. Now here it will be 2 to the power of 24. You understood? Because here he is not using pairs. He is saying you total chromosome. But as I said, if he says you pair, ignore the pair and take this 6 always. But if it doesn't
So,
give you, yes,
64 types of gametes or what? Or
yes,
combinations of
yes,
gametes.
combinations of gametes, possible combinations of gametes. Okay, next question please. We are going to applied genetics now. Which of the following is a true statement for the selective breeding?
All of the above.
All of the above. Very good. Please read them because it gives you new varieties. Offsprings with the desired characters can be produced, and also hybridization and inbreeding are the types of it. Okay. If desired trait is dominant, what is the possible of its genotype?
So it should
If
be C, right?
it should be C. Yes, Hussein. Can you explain why C?
So because just like even by reading the options, it says homozygous recessive, like the third option, it cannot be an option. And heterozygous could be dominant, and
Yes,
so does
yes,
homozygous.
very good. So this answer cannot be your option for the answer because it is homozygous recessive. But your desired character is what? Dominant. And this dominant, you can either take it from capital T, or capital T, small t. So these are your only two options from where you can take it. So this option is heterozygous and this option is homozygous dominant. Okay? Very good. Next page, please. A farmer observed the desired trait of a red rose, but is unsure of the exact phenotype. This is again a test cross, boys. You know the phenotype, but you don't know the exact phenotype. Sorry, exact genotype. So what you do? You do always a cross. A farmer observed the desired dominant trait of the red roses, but is unsure of the exact phenotype. What can he do to identify the genotype? So remember what was the definition of
C,
the test
mister.
cross? In the C, complete a test cross with homozygous dominant.
Should
Do
be
you do a test cross with homozygous dominant or homozygous recessive?
homozygous recessive.
Yes, you do a test cross with homozygous recessive. So the answer is D. Remember, boys, just now we did capital T, small t with small t, small t. You always cross it with small, small. This is what test cross mean.
Mister?
Yes?
But if it was homozygous dominant or heterozygous, the phenotype will be the same.
No,
So,
no, no. It will not be same. Try it. What you are saying as per option C, you are saying that he will choose capital T, small t, cross it with capital T, capital T. And what option D is saying, he will take capital T, small t, and he will cross it with small t, to find the genotype of the offsprings. So answers will come totally different. Over here, what answer will come? It will come, do the Punnett squares, capital T, small t, capital, capital. So you have capital, 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 small, capital, 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 capital. So you get 3 capital T, and only 1 capital T, small t. This is what answer comes from here. And this is the test cross. You know it already. It will give you what? 2 capital T, small t and 2 small t. So it will give you 1:1. Okay.
Can you go down?
Okay, let's go next, please. I don't think so there is any other question on this page. Okay, the last page, which is the basic patterns of inheritance. Boys, by the way, there are 6 diseases. I'm going to prepare, inshallah, a revision sheet that will be ready by tomorrow. Inshallah, I have made it my priority. I'm going to prepare a revision sheet from the book and from the presentations to merge everything and put it on one place, so that you can learn it from one place. So what you need to do, all keep checking the LMS, and in the LMS I have placed here CA1 term 1 revision and practice. It's not yet activated, by the way. Let me activate. It's not yet activated. It's now activated. So I will put the revision sheet here also. I think so. I need to refresh the page. So yeah, yes.
Can you activate for the health? Because if you activate for the health, the book of the health will be in LT1.
Really? No, this one doesn't have to do anything with Aldivan. I
Sir,
have yes.
When I raised the ticket, they said that the teacher should open the health sciences LMS.
Okay. Which one is your Hamdan? CAI?
CAINI.
Okay. CAINI. Okay. I will show you. Sorry, we are now at a different place. I have activated all the way all what is needed by you in Unit 1, Unit 2, and even in Unit 3. But Unit 3, I didn't activate it yet. I will do. We finished until Unit 2. Okay. It's activated in your class also. This is ENICAI. Okay. You can have all the presentations here, but this book, Unit 1 book, and there is no full book here, and we are till now we are not allowed you to give these PDF. You need to visit. Okay, I will activate this workbook, but you have to go to LMS. Sorry, Aldivan. Are you able to log into LMS?
Yes, Mr.
Okay, click here in the 3 dots. When you click here in the 3 dots, where is Aldivan? Here is Aldivan. Okay. When you go to LMS, click here on the 9 dots, click Aldivan. It will automatically take you to your book. You can search your book from this page. See, it's coming Aldivan now. Don't log into Aldivan directly. Log into LMS, click on these 9 dots, click on Aldivan. It will bring you here. Use your LMS email and ID. Same you use for the LMS. You need to use here same. When you use this, and it will take you to the library. Okay, let it come. It's coming. Wait. Yalla, faster. I don't know, it is stuck. By the way, it will take you to here, and you will have the health book here. But for the time being, you can use all the workbooks presentations. I have activated everything for you. You don't see them? That these are activated for you? Okay.
Mr. I saw it. I saw that there is health.
Okay, great. Okay, boys, now this is 1.4, the basic patterns of inheritance. We need to go it now. 6, individual I1, I2 and 2. Yani I2 and II2. What are these? Carriers. And who are the carriers?
I2.
Yeah, they are
the people that they look normal but they have a disease inside.
Yes, no, they have the allele for the disease. They don't have disease. Okay, they have the allele for the disease. So they are normal, but they carry an allele for the, okay, selective breeding. Here, see, an individual who is heterozygous for a recessive disorder. This is the simplest definition you have. This is in your book. An individual who is heterozygous for a recessive disorder is called a carrier. Okay, by the way boys, these tables are very important. In your book I will show you. There are, hey, these tables, this one, it's on page number 27. This has all 4 recessive disorders, and then page number 8, this has the 2 dominant disorders. No need to remember the occurrence, but you need to remember what is their cause, what is their effect, and how you can cure them. Normally no cure, no cure. Okay, and here you have mainly no cure in all, but there are some other cures. You need to see them. This table and the next table, these are important. I will put them in the revision sheet, inshallah. Okay, let us go back now. A is the answer. Okay, which of the following is not a recessive disorder?
D, Mr.
D. Yeah,
Hunting.
Huntington.
Yep.
Hunt. From Hunt. Huntington. Okay. In this pedigree, identify the pattern of the inheritance in the pedigree shown.
Mr. It's a Y-linked because female. I mean X-linked because females are
Mainly affected.
yeah. So it's
Okay.
dominant X-linked.
Yes, it is a dominant. It is a dominant X-linked. Very good.
Sir, but why is it dominant?
Okay, why it is dominant? Here it is very important, and remember it. Remember, I'm saying again, if it is a recessive, again, if it is a recessive, both of these parents should be heterozygous, or at least one heterozygous and one homozygous recessive. This is number one. You understood what I said?
So, sir, if it's recessive, then both parents are not gonna be carriers. Is that what you're saying?
No. If it is recessive, both parents should be carriers. Should be. And
So,
normally,
if the female is black, means it will be recessive.
no.
Recessive.
If all these colors which are shaded here, all these colors which are shaded here means they show the trait. Okay, it means they show the trait. Once again, boys, sorry. All these individuals who are shaded here, it means they show the trait. Yani either they are diseased or they have a certain character. We don't know, like, this pedigree is for what, because they didn't tell us this pedigree is for what. This is just as a pedigree. Okay, now they show a character, but this female is not showing a character. Now either this female is capital T small t, or this is a small t small t. You got it? Boys, are you there?
Yes, Mr. We got it.
Okay, and another thing that you can focus here, actually they didn't extend this pedigree, they should extend it. You see over here that they have some normal and some showing character, so it's a mixture, some normal, some showing the character. Okay, and they are not in 50%. And then they should have another thing over here saying that when they cross two normal, all whoever come normal, like this, all come normal, none of them come yani diseased. That proves you that this pedigree is a dominant pedigree.
Mr.
Yes.
The two X and X, they are dominant or one of them only?
One only, X dominant. One is only dominant. If two are dominant, then all the males will become all the males will become infected. If you look into the example, sorry, here, if you look into the example, this male is coming normal, right? This male is coming normal, this male is coming normal. It means that this pedigree,
Sir,
yes,
this both female that is dark. This is called recessive Y.
no, not recessive Y. Y is only present in the male. Boys, listen. If this is a dominant Y link, the recessive Y link, it is not in your topics. We did not study these two, and we are not going to study these two, dominant Y and recessive Y. So automatically, option C and D are wrong. First thing. Second thing, we are left with option A and option B, which is dominant X linked and which is a recessive X linked. If this is a recessive X linked, only few of the individual will be affected, and mostly both parents will be showing as normal, but they will be carrier. Understood or no? Wait, let me show you quickly a recessive X-linked pedigree. Then maybe it will be easy for you. Here, wait, recessive X-linked disorder. Yes?
In the generation 2, the males are carriers or what?
In the... Wait, 1 minute. I will go back there what you are asking. But let me see a pedigree first. Here, this one. See. This is an X-linked disorder. So see here, if it opens, okay. No, it's a journal. Okay. Okay, this is an X-linked disorder. Now here, if you notice, here the male was not infected in first generation. But when the two normal, they look normal. So they are normal, right?
Yes.
They are normal, but they get a male who is diseased. Understood?
Yes.
Now how this disease comes, because here it is normal, but this is carrier. The X which came in the male, come from the mother. Remember always, the X which come in the male come from the mother. So what come down? The mother was heterozygous, so the come down as X, and that is a recessive X, and it got it infected. But when you look here again the male is getting infected. The female is not. You remember we studied that there is no carrier in the males. We studied it, you remember? But all the females you see, most of the females which are following in this pedigree are carriers. They look normal. So this is how you identify that only few females, if only few females are infected. How many females are infected in this pedigree, boys?
One.
One only, sir. And how many males are infected?
4.
1,
Three,
2,
Mr.
3. Okay. Look, there are more males infected as compared to the females. It means that this pedigree is a recessive pedigree, because the male cannot be a carrier, but all these with the dots, this dot, this dot, this dot, all are carrier. This dot is showing a carrier in the female, because the female needs two alleles on both X chromosomes to be diseased. Now look back into your question here. Here you see that more females are infected. It means that the disease was dominant. Is that clear now? Because
Mr.
even if only one X comes as a dominant in them, for example, if it was XT and X small t, now if this is a dominant disorder, will she a diseased or a heterozygous?
Heterozygous.
لا مش heterozygous boys. Listen first. I said that disease is dominant disease. Yani this capital, wherever it will come, it will show that this is diseased. Now this female is a diseased or heterozygous? She is diseased. She is heterozygous. Fine, because you are looking at capital T small t. But she will be a carrier or she is diseased.
Disease.
Diseased. Very good. Now you understood. But if the disease is recessive, now I'm going to the second question. This here. If the disease is recessive, now this female is a carrier or diseased?
Carrier.
Carrier. Very good. Very good. But this is not the case with the males. Okay. Now the male, if it is X small t and Y for the same type, tell me if the male is diseased or normal or carrier.
Disease.
Diseased, because male need
Diseased.
only yes, very good. I hope you understood. No worries, read it more. If there will be any question, we will go over these. Last question, boys. What does a pedigree chart with only males affected and no female affected or even carriers indicate?
Dominant.
Y-linked
Yes,
pattern.
very good. It's a Y-linked pattern, but we have not required it anymore. But anyhow, still choose the answer C, which is a Y-linked pattern, because only males are affected. No female and no carriers are indicated in that. So this is a Y-linked pedigree.
Mister,
Yes,
can you please remember me again? That the X is female.
yes.
The X is female.
XX is a female and XY is a male.
No, I'm asking you a normal question. That is X is female or male?
What X? Yeah, okay. Sorry. Yeah, X-linked, definitely it is coming from the mother, the X-linked characters, because the male gets its X from the mother.
Oh,
So,
okay.
because see, this X in the male always come from the mother, and Y come from the father. Okay, so this one is coming from father, and this one is coming from mother. Over here, this one is coming from father and this one is coming from mother. Okay, so whatever disease is in the mother and it is located on the X chromosome always show in the males, but not in the females, because females can be carriers but no male can be carrier. Okay, thank you
Mister,
so much. Yes.
can you go back
Could
to
you?
slide 9, question 10?
Slide 9, question 10. Anyone else who joined late and I did not put his attendance? Yes, please tell me which one. Slide.
Sir, here they didn't say that offspring, so how I know if they are talking about the parents or the offspring?
A farmer observed the desired dominant trait of a red rose but is unsure of the exact phenotype. What can he do to identify the genotype of the red rose? Here, since ignore everything, since you see this statement, what can he do to identify the genotype of the red flower? What is the phenotype here?
The phenotype is the red flower.
Red. Okay, now listen carefully. I'm going to state for you the definition of the test cross, and that definition says that a cross that is made to find the genotype of a known phenotype. Known phenotype. Okay, now use this definition back up here in this question. What he can do to identify the genotype of the red flower, a cross that is made to find the genotype. What he can do, so he can do this cross. And what is the name of this cross? A test cross. Okay, so again, what he can do, he can do a test cross. You got it? He can do a test cross. And what is a test cross? I have already listed here, capital T, small t with small t, small t. So he had to complete a test cross with a homozygous recessive. So this is the homozygous recessive individual.
Sir?
And this is the one which you are calling as red. So he took that red and he did a test cross. Yes?
Can it be homozygous dominant with this?
Yes, you are correct. Yes, it can be, because we studied two cases, Hamdan, you are right. Because I focused only over here with one. You are correct, Hamdan. We studied two cases of the test cross. If you remember, when we were studying test cross, okay, so we said that test cross can be done, case 1, case 2. So in case 1, you can take capital T, and always cross it with small t, small t. But in case 2, you need to take capital T, small t, cross it with small t, small t. Okay? Now you will focus on the individuals. If 100% dominant comes, it means that the parent was homozygous dominant. Okay, and if they come 1:1, it means that the parent was heterozygous. Is that clear now?
Yes, Mr. Mr. Can you send me the code of this class click, because I cannot see your notes.
Okay, what I will do, because my notes are on my page, I'm using my own name. So yes, my notes are here in my name. Okay, wait, what I'm going to do, 1 minute. Okay, download, I will do it now because later I will forget. Export student work to PDF. Okay, 1 minute. See, I need to export my own work. Muhammad Usman Farooq, this one. Export. Because I'm working on my own name, so reflecting you through second screen. So I will export my own. It's taking time. It should not take this much time. So once it is exported, I will put, it's done. I think so. It's done. Yeah. Wait. My classes, biology, question bank, and here practice sheet for CA. Save. Let me upload directly into LMS, and I will upload directly into LMS. Why it's not coming? CA biology, and we have here CA1 practice question. Okay, this one. So I will add this here. File, add. Okay. Which one was this one? This is with the answers that shows my name. Okay. So this is practice questions with teacher's notes. Now I will make it for everyone in all the thing. Right now CAI can see it, but now I'm going to put it into CAI ENI and also in the ENI. Okay, you can see it there in the LMS. When you go there, see CAI term 1 practice questions. Okay.
Sir,
yes,
could you please go to page 5? I have a question, please.
boys, the others who don't have the question, thank you so much for your day. All the best, and keep checking LMS CA1 practice questions because I will be putting all there. Yes.
Mister, how you get 1/2?
How I get?
How you get the test cross the ratio 1/2?
How many these are total, Salim?
Mister, I think it is 1/1 because
How
as you
many
said,
these? How many these are total, Salim? Total.
50%.
Yes. What does 50% mean? 1/2. Because here we are checking the probability of the dominant only. We are not comparing the dominant with the recessive. We are
What
just.
about question?
What about question what?
Question 10. How you get it is the 3/1?
Because this is the phenotype of the Mendel's F2. What is F2 when you cross two heterozygous? Cross two heterozygous, capital T small t crossed with capital T small t. What you will get? 3/1.
Mister, I cannot see your drawing.
I'm not drawing it. I'm just saying it to you. You can do it yourself. Capital T small t crossed with capital T small t. Take two heterozygous as we do it in F2. Monohybrid cross. Salim, monohybrid cross, my dear. Go to chapter 1. This is the monohybrid. Hey, this one. This is what he is asking you. You need to find F2, the phenotype of the F2. So, how you get F2 when you do F1 with F1? What is F1? Heterozygous. So cross heterozygous with another heterozygous, you get all these individuals. Now look at their phenotypes. 3 are yellow, 1 is green.
Mister, if they ask us the hybrid.
Dihybrid, then you need to look for this one. This is dihybrid. The dihybrid ratio of the F2 is 9:3:3:1.
Oh, okay, Mister. Thank you so much.
Okay? And for the dihybrid, the genotype and phenotype ratios both are same, no change. But in the monohybrid, the F2 genotype ratio is 1:2:1, and the phenotype is 3:1. Here, it's written here. If you look into the book, you have all the answers.
Yes, Mister. Mister.
Yes, please.
So for page 4, question 2, I really didn't understand it. Only this question and finish.
Yes, there was another one. What do you have?
Page 4, question 2.
Yeah, this is page 4. This is question 2. What you need?
Mister,
How we get 1:1?
Yes, Mister.
Salim, he did a short with long. What is the result?
Mister, when you why in the short you put two
Salim,
capitals and
Salim, just answer my question, please. So only you will be able to understand it. Don't ask further questions. I'm asking you, when he crossed a short with long, what is the offspring coming?
long joined.
It's not coming long. Salim, read the question. Read question, yalla. Ikra.
A short-toed animal was crossed with a long-toed animal of the same species. All the offspring had a short
Okay,
toe.
stop here. Stop here. When he
Yes.
crossed the short with long, all the offspring came what?
T capital with small capital. Small t. Small t.
Salim. Habibi, just tell me whether they come short or tall, short or long.
Mister, I think it is tall.
Why you think? It's written here,
Oh,
Salim.
Mister, sorry, Mister, sorry, it is short.
Salim, don't think anything when the information is given you in the exam. Here the information is given. Don't think. They say that when you cross short with long, all the offsprings are short. Halas, story finished. Same here. Short, long, when you cross them, all came short. So who is dominant?
Short.
Halas. Until here clear? Now they say that they crossed this short, they crossed this short with long. So your short which it came was capital S, small s. And they crossed it with long, who was recessive. So small s,
That is
small
to
s.
the
Yes. Do the Punnett square, you will get answer 1:1.
yes.
Okay, when the information is given in your exam, please don't use your own thinking, then you will get wrong answers. If the
Can I
information is
screenshot it? Can I screenshot it?
this
Can
one,
you
I
read the
already
page?
this one is already in the LMS, my dear. Do the screenshot,
Mister,
it's okay, but I put already in the LMS.
Mister,
can
Yes,
you
please.
can you let it big?
Yes,
Screen
Hamdan.
big.
instead of the question in the exam about the F2 generation, they will say if it's dihybrid or
Yes,
monohybrid.
yes, they will say you dihybrid or monohybrid. If they don't say you anything, you need to work for monohybrid.
Okay,
Is
Mister.
that clear? If they want specific one, they will say you monohybrid or dihybrid. If they don't say anything, it means that they are talking about monohybrid. Okay?
Mister,
Yes, Salim.
no problem. No problem if I ask you another question.
Yeah, the faster, boy, my class was from 1 till 2. You can see the time, 2:40. I have appointment at 3, honestly.
Okay, Mister, only will take it,
Faster,
this
yalla,
question.
yalla, faster.
The question after it, question 6.
Huh?
In one type of plants, the allele for red flowers is dominant to allele for the white flower. A plant with a red flower is crossed with a plant with a white flower. Half the offspring have a red flower and half white flower.
Half mean 1:1.
Yes, Mister. That
1:1.
15%.
Okay, that comes only following red the test cross. And what you do in the
Oh,
test
Mister,
cross?
means I use this all. First one is I don't think so. It is correct. The first one.
Yeah.
I will use the B and C and D and test cross, and I will see what is has a 50%
Yes.
and 1/1,
Yes.
and I will get the
Yes.
answer. Yes.
Yes. Yes. Yes. True.
This is a good way. Yes.
Yes. Exactly. Thank you so much, boys, and I'm much
Thank you, Mister.
appreciated for all your interest and coming. Enjoy your day, and inshallah, wish you all the best for your exam. If anything you have, you can email. I will answer. Okay?
Okay, Mister. Thank you so much.
No problem.